Okay, rapid onset obesity with central hyperventilation, hypothalamic dysfunction, autonomic dysregulation, Rochat syndrome is a rare disorder characterized by respiratory failure, autonomic deregulation, and endocrine abnormalities. Some of these patients could also present with neuroendocrine tumors, adding the net prefix to the acronym. The diagnosis is based on clinical criteria defined by Ice-Ludlot in 2007, and the main criterion is rapid and sudden weight gain associated with central hyperventilation in a previously healthy child. Upon diagnosis is based on early recognition and is essential to provide timely respiratory support and to minimize morbidity and mortality. The pathogenesis of this syndrome is still unknown. However, there are three main hypotheses postulated. First one, the genetic theory. Many studies rule out candidate genes, including FOX2B, that is the gene responsible for the congenital central hyperventilation syndrome, and other genes that are involved in the function of the hypothalamic, neuroendocrine, and autonomic system. The epigenetic theory based on the report of discordant presentation of Rochat syndrome in monozygotic twins, and finally the immunology that is based in reporting of Rochat patients in whose cerebral spinal fluid analysis disclosed an intrathecal synthesis of oligoclonal bands, and anti-hypothalamus and anti-pituitary antibodies were detected. Also, the association that is reported with celiac disease, and finally, the positive effect of the intensive immunosuppressive treatment that are reported in some cases. Regarding the acronym and the clinical presentation, the rapid onset obesity is the first recognizable sign with a median age of 3 years. Hyperventilation, that is the most life-threatening feature with a median of 2.2 years appearing before the onset of weight gain. The hypothalamic dysfunction that appears rapidly after onset obesity, being hyperprolactinemia, the most frequent described, autonomic disorders, the neuroendocrine tumors that are present in 50% of the patients, generally the ganglioneuromas, ganglioneuroblastomas, and being the adrenal the main location. The neurobehavioral disorders, although they are not described in the acronym, are a very characteristic sign of the syndrome, and others like metabolic syndrome, insulin resistance, celiac disease, scoliosis, and dyslipidemia. The aim is to describe the clinical biochemical features, treatment, and outcome in a cohort of pediatric patients with Rochat syndrome, followed in a single tertiary center, and to compare our data with that reported in the literature. We conducted a retrospective review of the medical records of 10 patients with Rochat syndrome followed between 2007 and 2021. And the clinical data were first analyzed individually and then compared with the cases reported in the literature. Results are presented as mean SD for quantitative variables and as a percentage for non-quantitative variables. Regarding the clinical characteristics of our patients, we can see that the mean age of diagnosis is 7 years, but the onset of weight gain appears approximately 2 years earlier with a mean age of presentation of 5 years with a mean SDS-BMI of 5.4. Hyperventilation, the second hallmark of this syndrome, is diagnosed at approximately 7.5 years. All our patients have a hyperventilation. 70% had central hyperventilation requiring non-invasive ventilation. Between these two symptoms, hypothalamic pituitary disorders and auto-dysregulation appears. Neurobehavioral disorders appears at an early time coinciding with the onset of weight gain, and tumors of the neurocrystal origin also have a median age of diagnosis of 5.4 years. These are the height and weight curves of our population. We can see a color line drawn for each patient, each female patient, and we can see a clear tendency of weight gain associated with a decrease in growth rate. That occurs in contrast to what happens in exogenous obesity that is associated with accelerated growth. We only have one male patient that presented with the same pattern. Here is the diagram for each specific group of symptoms of our patients. This is a box plot where we have the median and the first and third quartile, and the T bars that shows the rest of the data. Here we can also see that the rapid onset obesity, the neurobehavioral disorders, and the neuroendocrine tumors are the first signs to appear, followed approximately 2 years by the rest of the symptoms. Here we can see the age-at outcome of hypothalamic disorders. As you can see, all the symptoms appear before 12 years of age, except for one patient who was diagnosed with adrenal insufficiency at an older age, and of course, hypogonadotrophic hypogonadism that is diagnosed at a later age. Here's the comparison of our currently reported cases and our series. We take into account the last review published in JCM 2020. They have 43 patients and our cohort of 10 patients. We have a female preponderation, no mark in our series, the rapid onset obesity, the hypoventilation, the neurocrestal tumors, and neurobehavioral disorders were similar, but we tend to see a more frequent in the hypothalamic pituitary dysfunctions. Regarding the mortality, we have a higher rate of mortality, but we also have a long time follow-up with a maximum of 12.5 years. Indeed, one of our patients died in adulthood. In the literature, the reported mortality is low, but they don't have many information about follow-up time. So to conclude, this is one of the most large series of patients with ROHAD or ROHAD-NET syndrome that are followed in a single tertiary center. Sudden weight gain associated with neurobehavioral disorders and neuroendocrine tumors should raise the index of suspicion for ROHAD and lead to search for other comorbidities of this syndrome. Although the clinical characteristics of our patients match with those described in the literature, we presented a higher frequency of hypothalamic pituitary disorders. Early recognition and evaluation of the syndrome facilitates the management and reduces mortality of these children. So the complexity of this entity and its high mortality rate requires strict long-term follow-up by a multidisciplinary team. And finally, a better characterization and a large cohort of patients are needed to increase the knowledge regarding this syndrome and this entity. Thank you very much.
Gracias, Mariana. Se abre la discusión sobre el paper. Rodolfo.
Buen día. ¿Preguntas en inglés o en castellano?
Como quieran.
Como quieran, en castellano. Mariana, bueno, felicitaciones por esta serie tan grande de algo tan poco frecuente. La pregunta mía es por la observación de una mayor frecuencia de trastornos neuroendocrinos. ¿Podrá ser esto debido a que son pacientes derivados a un servicio de endocrinología y que eso haga que tengan mayor proporción?
Sí, nosotros creemos que tiene que ver con que, bueno, en el hospital, al tener varios pacientes seguidos, muchos estuvieron internados en las salas de pediatría. También tenemos un equipo de nutrición que sigue a estos pacientes y la unidad de sueño. Entonces, hay una derivación rápida de estos pacientes que consultan por obesidad, tanto al servicio de pediatría como nutrición, a nuestro servicio, donde nosotros enseguida evaluamos toda la función hipofisaria y creemos que hay un bias con respecto a eso, de la alta frecuencia que encontramos de estas alteraciones.
Doctora Chiesa.
Felicitaciones, la verdad que muy lindo y un gran aporte porque estos chicos andaban dando vueltas y atendidos. En tus conclusiones ponés que una intervención temprana y un seguimiento adecuado los mejora. Quisiera saber, porque seguramente no fueron todos simultáneos, sino que me han ganado experiencia en el tiempo, si ustedes todavía están viendo un camino para tener en estos chicos o si ya pueden aproximar qué es lo que rápidamente uno debe hacer y debe evaluar con rapidez para intervenir.
En realidad lo que nosotros creemos es que en estos chicos la obesidad comienza después de los 2 años de vida para descartar otras causas de obesidad de inicio muy temprano, y lo que nosotros vimos es que rápidamente aparecen trastornos de la conducta que deben ser evaluados con un interrogatorio dirigido porque si no muchas veces puede pasar de alto. Entonces rápidamente entre estas dos asociaciones buscar la presencia del tumor, que es lo otro que aparece temprano, y rápidamente derivar a la unidad de sueño para la evaluación de la función respiratoria, que es lo que determina la mortalidad de estos chicos.
Gracias.
Mariana, una pregunta sobre la mortalidad. Tienes un 30% de mortalidad. Cuéntanos un poco de qué fallecen estos pacientes y cómo podemos tratar de reducir esa mortalidad.
La mayoría fallece de causas respiratorias. Nosotros, de las 3 pacientes que fallecieron, una falleció en la vida adulta, que la mamá se comunicó con nosotros para informarlo, pero no tenemos mucha información, y 2 pacientes fallecieron en la casa durante el sueño, así que se cree que tuvo que ver con una alteración de la parte respiratoria y es por eso que hacemos tanto hincapié en rápidamente ante la sospecha derivar a la gente de neumonología para que tenga un buen soporte de la parte ventilatoria.
Claro, como tener algún sistema de alerta sobre apnea nocturna.
Sí. Ellos tienen esos sistemas de alerta cuando baja la saturación, pero bueno, una paciente falleció en la casa y en realidad esto fue analizado retrospectivamente cuando se la llamó para citarla a un control y la madre mencionó que falleció y otra falleció internada en una institución.
Ok, gracias Mariana.
No,
Vamos al
nada.
segundo paper.
Bien, el segundo trabajo va a ser presentado por Ana Benedetti de Brasil, es Identification and In Silico Characterization of the PL713P Variant Activation Function 2 region of the androgen receptor gene in a partial androgen insensitivity syndrome patients.
Good morning. First, I'd like to thank the scientific committee of this meeting for the opportunity to present our study. It was performed as a collaboration between the Universidade de São Paulo de Ribeirão Preto, and Universidade Federal de Alagoas. There are no conflicts of interest to declare. The androgen insensitivity syndrome, or AIS, is the most common etiology of 46XY DSD, and it is divided into 3 phenotype groups. The first group is the complete form, or CIS, where the patient presents typically female external genitalia, gonads developed into testes and breast development. The second phenotype is partial, or PIS, and it also presents with a typical external genitalia, gonads developed into testes and breast development. And finally, the mild type, or MIS, where the patient presents with typical male external genitalia, gonads developed into testes, and male infertility and organocomastia. The phenotypic spectrum of AIS is explained by the residual activity of the androgen receptor. The androgen receptor gene is located at X chromosome and has 8 exons. These exons code for a protein that has 4 different domains, and exons 4 to 8 code for the ligand binding domain that is important for protein function. This protein functions when it binds to agonist testosterones and beta-testosterones, and it will regulate the expression of several genes that are important for male gonads and genitalia development and secondary male characteristics. AIS molecular diagnosis in the AR gene is around 90% for CIS patients and 50% for PIS patients. Several mutations have been described throughout the whole gene in the 3 phenotypes, especially around 600 to 900 amino acid position, that is where the ligand binding domain lies. Inside the ligand binding domain, we have the activation function 2 region, or AF2. This region is composed by 11 residues highlighted here, and it's where the agonists bind to the protein. So the objective of this study is to characterize a novel AR variant located in the AF2 region found in our AIS cohort, and to search for other variants in the same region in individuals with AIS in the literature. So first, we searched for mutations in this region in the literature, and we could find 6 variants that were mostly related to PIS and 1 related to CIS, and 3 out of 6 of them had a functional assay. The variant found in our cohort was a change of a leucine to a proline in codon 713 in a patient presenting with CIS phenotype. In silico prediction algorithms, all predicted it as to be deleterious. To further analyze this mutant, We downloaded from Protein Data Bank a crystal structure of the LDB in complex with DHT. Then we used modular software to repair the missing residues and atoms, as well as to create a single point mutation, leucine 713 proline, using this structure as model, as we can see here. Okay, so molecular dynamic simulations, we performed it using ROMAX with charm 36 as the force field. The production phase was at 300K for 100 nanoseconds, trajectories were saved every half nanosecond, and ligand parameters were obtained by the Sesion FF server. The system was equilibrated in 2 steps, a canonical NVT ensemble followed by an isothermal isobaric NPT ensemble. So, to compare both complexes, we calculated the root mean squared deviation, which is called RMSD, and it calculates the average distance between the backbone atoms of the complex. So here, we can see in this graph that both wild type in green and mutant protein reach more stability after 30 nanoseconds, which is very different from the highly unstable before. Also, we can see that the mutant complex has a trajectory that's more unstable in comparison to wild type. During 40 to 50 nanoseconds, we can see that both complexes had an RMSD value above 0.2 nm. Okay, so for the ligand, we can see here that they reach stability after 50 nanoseconds, both the wild type complex and the mutant. The mutant is more stable until around 80 nanoseconds, where it reaches a slight increase. Both of them also reach stabilization at around 0.15 nm, which is lower than the 0.2 value, which shows that most likely the ligand with the binding of the protein, it's not going to have much more variations than this. So to conclude, clinical evidence of the AF2 variant suggests that its disruption is not always related to AR abolishment, as most AF2 variants cause PIS. The novel protein we found, L713P variant, was not able to massively change protein structure. However, it needs higher energy to sustain the DHT AF2 binding. Also, this suggests that AR residual function can remain even when there is a disruption of such an essential AR region. I would like to thank the DSD team at our hospital, especially Professor Berenice and Dr. Rafael Locke, also the postgraduate student Raquel, and both of our collaborators, Dr. Reginaldo Petroli and Professor Silvana Gilatti. Thank you very much.
Muchas gracias, doctora. Queda abierto a preguntas.
Parabéns pelo trabalho.
Gracias.
¿Se escucha? Yes, ahora sim. Parabéns pelo trabalho.
Obrigada.
A pergunta é, você sabe a consequência possível desta diferença que você mostrou na dinâmica do receptor?
A consequência possível seria de que ela precisa de mais energia para se manter, né?
A consequência da dinâmica no receptor no binding to DNA or trafficking to the nucleus?
I'm sorry, I couldn't understand.
Okay, I start again in
Okay.
English, maybe. the consequence in the dynamics of the receptor, either, for example, in the nuclear translocation or binding to ARE?
Oh,
Is it known? I don't know. I'm ignorant. I'm asking.
I'm not sure. Actually, I think this is a very interesting question that I'm definitely going to ask our collaborators for us to do. Thank you very much.
Thank you. Congratulations.
Thank you.
I have also a simple question following on Rodolfo. Is this increased energy that's needed for the binding, just binding affinity or binding capacity?
I'm sorry, could you repeat it?
You show that the mutant needs more energy.
Yes.
So the binding to the receptor
Yes.
is lower affinity because it needs more energy to
Yes.
do that, or is the binding capacity?
I believe it's affinity.
Thank you.
Muy bien, pues muchas gracias, doctora. Felicidades.
The next presenter. Now, the next presenter is Jonathan Kraus from Chile, and the topic is the Pacak-Zurhang syndrome report of a case.
Hi, good morning. Thank you very much for the scientific committee for this opportunity. I will present this case of this syndrome that I have to recognize that I met him when we saw this case. As an introduction, we know that pheochromocytomas and paragangliomas are tumors derived from the neural crest-derived cells from the adrenal medulla in the case of pheochromocytomas, or the sympathetic or parasympathetic paraganglia in the case of the paragangliomas. And with advancements of the genetic techniques, nowadays we know that around the 70% of patients with these tumors have a genetic background, approximately half of them coming from somatic mutations, and the other half coming from germinal mutations. And they are classified into 3 clusters regarding their physiopathology. The first cluster is associated with the pseudohypoxia pathway. The second, the cluster 2, coming from the WNT, from the kinase signaling pathway, and the cluster number 3 coming from the WNT cluster. Regarding cluster 1A, as you can see in this figure, is subdivided into cluster 1A and B. Cluster 1 is related to the Krebs cycle, and the cluster 1B is related to the hypoxia signaling pathway. And in this case, the gene HIF2A is related to the hypoxia pathway. First of all, under the activation of the VHL gene over here, the HIF2A is ubiquitated, and then it's degraded through proteasomes. And after the abnormal Krebs cycle production of different metabolites, and the HIF2A is diminished in its hydroxylations, it is unstabilized, and in that case, it diminishes the different DNA genes signaling, stabilizing different messengers associated with angiogenesis, metastasis, and proliferation. In cases of, for example, VHL mutation or different mutations here in the Krebs cycle, or in the same, or in mutations on the HIF2A genes, this product is stabilized and promotes more products that favors angiogenesis and tumor formation. What has been described from the HIV2A mutations, that the patients that have them, they are most de novo, they have a high metastatic risk, they have a neurogenetic profile, They have consistently hypertension beside more basic family symptoms. Though IHIF2A gene was known as a gene causing congenital polycythemia, it was on 2012 that the group of Dr. Karl Pasek in the NIH discovered the relation with polycythemia, and 1 year later it was related with pheochromocytoma. And as you can see here, these are a little cohort of 6 patients follow up for 6 years, and what they have in common, they have no family history, they have a polycythemia at an early age, all of them had a neurogenetic profile, all of them have paragangliomas at a median of 15, 17 years, and somatostatinoma at around 30 years. All of them had ophthalmological abnormalities, and I had a high prevalence of multiple disease, metastatic disease, and recurrency. So the objective is to describe a clinical case of a polycythemia, pheochromocytoma, paraganglioma, and somatostatinoma syndrome due to a known HIF2A gene mutation that we study in our center. This is the case of an almost 18-year-old male, full-term AGA with normal perinatal history, born to non-consanguineous parents that were healthy. He had a normal growth and puberty and normal neurodevelopment, and he had a diagnosis of a polycythemia since 2 years that it has been managed with recurrent phlebotomies. He had no etiological diagnosis until this time, and at the age of 10 years he had a medullary infarction with a secondary enuresis due to that. And at about 16 and a half years, he had a diagnosis of hypertension, managed with enalapril and carvedilol, and occasionally he had some sweating and palpitation episodes. And an out-center study showed a high urinary norepinephrine study. So he was admitted in our hospital with a very high blood pressure and with a suspicion of pheochromocytoma. He had a normal physical exam, and he was on complete puberty, and he started with doxazocine, and he had a normal nephrological, ophthalmological evaluation, and normal echocardiogram. Unfortunately, in Chile, we don't have a plasma fractionated metanephrines, but we were able to measure them on urine, and you can see here the high level of normetanephrines. And what we had in the very beginning, it was abdominal urease, where you can see a right adrenal nodule with low vascularization. Due to the suspicion, the patient had a gallium dotatate PET CT. In Chile, we don't have F-DOPA, that it was the imaging of preference in this case. And he had bilateral adrenal nodules with a moderate to intense captation of the chroron contrast, and also bilateral retroperitoneal adenopathies. Considering that, we suspect an HIF2A mutation. So after standard preparation, the patient went into a partial bilateral adrenalectomy and lumbo-aortic lymphadenectomy. He started on adrenal hormone replacement, and he had a rapid normalization of blood pressure. A couple of weeks later, we evaluated the adrenal function, and as we can see, he had a subnormal ACTH response for cortisol, and he had a pheochromocytoma paraganglioma panel that was negative for all of these genes. As you can see here, this is the patient's histology, the normal adrenal gland with typical nested cells of the pheochromocytoma, the capsule invasion, the lymph node invasion, and also positive staining for synaptophysin and S-stins that show a crestal neural origin. And they have 2 scores that are related to high metastatic potential. And considering the multiple cases published in the genetic lab, the exon 12 of the HIF2A gene was sequenced, and we found a C2T change in position 15A 89 that can change an alanine at the 530 colon to valine. That's the same mutation reported in previously papers that I showed. Unfortunately, he had a remnant of one of the adrenal beds, so we went into another surgery, and after that, until now, he has been with no symptoms and normal urinary metanephritis. This is the follow-up we are planning, though we have been doing it a little bit faster, and what is recommended in recently reviews. And we are pending the screening from somatostatinoma. These are the two questions I have for myself. How do we screen for somatostatinoma? With imaging, somatostatin levels that we don't have in Chile, symptoms, maybe IGF-1, considering the negative effect on growth hormone, and a question that I still have a little bit brave question: if we should left this left adrenal remnant, considering the highly potential metastatic of this issue. And these are my conclusions. We should screen for HIV gene in case of polycythemia, idiopathic polycythemia. We should start early screening for pheochromocytoma paraganglioma and later somatostatinoma, and the big relevance of genetic testing in all patients with pheochromocytoma or paraganglioma. Thank you, and I apologize for the time.
Hugo? Congratulations, it's a very interesting case. As you know, there are no international guides to follow for the apparition of somatostatinoma, because many cases are appear without symptoms, and perhaps I suggest to add to your suggestion the medication of C-peptide, glucagon, and also every time you measure IGF-1, you could measure GH to correlate the levels and to see if GH is every time abolished.
Perfect.
It's just a
Thank you for the suggestion. We can measure C-peptide and GH. Thank you very much.
Fernando. Go.
Gracias, Jonathan, por tu presentación. And we're going to move now to the fourth talk. And the fourth talk this morning is tumor DNA methylation profiling is a comprehensive prognostic marker for pediatric patients with adrenocortical tumors by Sonira Antonini from Brau Preto, Sao Paulo, Brazil.
Okay. So good morning. First, we, on behalf of my group, especially Ana Carolina Bueno, my postdoc, who can be here today, I would like to thank you for the Scientific Committee for this opportunity to present this work. As you may know, in Brazil, we have the highest worldwide incidence of pediatric adrenocortical tumors, PST, from now on. And this highest incidence is related to the presence of germline founder P53 mutation, the R37 age mutations. And due to the high incidence of this disease, especially in Southeast and South Brazil, we get a chance to study in details more etiopathogenesis and also the follow-up of these patients. So in children, adrenocortical tumors are usually functional tumors. Our patients present with signs of hormones, excessive hormone secretion, namely, especially hyperandrogenism, starting with precocious puberty and then progressing viralization. And from a simple histological point of view, most of these tumors are classified as carcinomas. For those patients with unlocalized disease, adjuvant systemic chemotherapy has so far limited improvement in survival. Regarding progression, fortunately, most of the patients have a good outcome, but for those patients with residual or metastatic disease, the 5 years survival is lower than 15%. With regards to aggressive disease and progressive prognostic features, aggressive disease is more frequent in patients diagnosed older than 4 or 5 years of age, in those presenting signs of hypercortisolism, and also in those patients in which the histopathological criteria, namely, especially those by Vinick criteria, are carcinomas, and with stage 3 and 4 are classified according to Sandrini's stage, now modified by IPI-CTR group. But today there is no robust prognostic or predictive marker. DNA methylation is an epigenetic mechanism that is a chemical change that changes the cytosine residue in a methyl, and usually occurs in CpG nucleotides within the CpG islands. And this epigenetic mechanism is usually used by the cells to prevent the transcription by taking the cells to the off state. More recently, global tumor DNA methylation profile has been used to diagnosis, especially for prognosis, in order to find predictive markers in several types of cancer. Therefore, the aim of our study was to investigate the association of DNA methylation in PST, presentation, and patient's prognosis. We evaluated 57 pediatric patients, most of them, as expected, were girls. We performed a global tumor DNA methylation profiling using this process, which allows us to evaluate over 850K probes. And then we processed data processing, quality control, and filtering, and we ended up to evaluate over 766,000 probes. Then we performed an unsupervised analysis of methylation data. Further on, we searched for association with disease features and patient's outcome, and to identify differentially methylated probes or DMPs. And then we performed 2 proof of concept studies, a candidate gene validation by qPCR, and a replication of our study in an independent cohort of 48 PST. Our first important result here is the unsupervised analysis, which means we analyzed only the methylation profile. There was no other feature of the tumors were known. And you can see that by using this unsupervised analysis, we were able to find 2 supergroups of tumors. The small one in blue, composed by 10 tumors, and the large one composed by 47 tumors in orange. We then compared the differential methylation profile between these two tumors, PST1, the small one, and PST2, the larger one. And here you can see the significant statistics that we adopted, -10 minus 6. And here is the differential methylation between the two. We found, using this very stringent criteria, 624 differential methylated probes. 418 probes were hypomethylated in PST1 group, and 224 probes were hypermethylated in the PST2 group. Overall, the group 1 was more hypomethylated, and we can see here these results, that over the PST group is more hypomethylated in blue than the group 2. But then we analyzed it in a more detailed, where these hypo and hypermethylations, CpGs, were located. In spite the PC group 1 being more hypomethylated as a global, we found out that in regions that were clearly associated with transcriptional regulation near to the transcription start site, there was more hypermethylated probes in the PST1. And here is shown here, hypermethylation in regions linked to transcriptional control in PST group 1 as compared to group 2. We then analyzed, we select the topmost hypermethylated gene in PST group 1, the small group. And this gene is SCAAP4 gene. This is a new gene in adrenal. And we don't know yet what is the meaning of this gene. But we see here that in all the 4 probes are clearly hypermethylated in PST1 as compared to PST2. You can see here that it is a clear differentiation. And then we analyze it by a transcriptome analysis, the whole mRNA expression. We see that there is a consistent association between hypermethylation of these genes and low mRNA expression of these genes in the same tumors. We then validated, as I told you, our data in another cohort of 48 patients with data that were publicly available in this former publication by Clay et al. And we were able to see the same results, 2 subgroups, and the same profile of hypermethylation in regions related to control in the PST group 1. And moreover, we built this oncoprint. And we can see here that in this figure that PST1 in blue is clearly associated with higher age diagnosis, clinical presentation with hypercortisolism, with advanced disease stage, and also with the need for chemotherapy and with this death. More patients were dead in this subgroup. But to be sure of that, we then perform multivariate analysis, and we confirm that PST methylation signature is indeed independently associated with patient prognosis. We can see here that patients in the group 1, the small one, the blue, had significant lower progression-free survival and significant lower overall survival. And these were even when adjusted for these co-founders. So the main conclusions in our study there are 2 PST methylation signatures, and they were associated with disease representation and patient clinical evolution, even in the context of a germline p53 mutation. And tumor methylation profile, according to our data, is a robust and independent prognostic biomarker for pediatric patients with AST. And I need to thank all the team that worked, including the bioinformaticians and the postdoc, Ana Carolina. But in these hard times that we are going through, it's very important to say that without this financial support of São Paulo State Funding Agency, FAPES, we would never have done a study like this and others in our laboratory. Thank you very much.
Thank you, Sunir. We've got a question there from Alex first.
Great talk, Sunir, great study. Let me ask you something. You show that this methylation profile is independent of P53 status, but do you check the CHAF1 genotype that is proposed as a modulator of phenotype of P53?
Yes, there is no data. I didn't show the data. We already genotyped the XAF1, and it's not published yet, but I can tell you that the results are a little bit different than it was published in the first study. It's probably, but we did not perform an association study with methylation, you are right. But in our preliminary results, XAF1 has nothing, is not that important, at least in pediatric adenocortical tumors. It's different for sarcomas as the original publication, but it's not published yet, so.
Alicia?
Oh, nice work. Congratulations. I would ask you about the sex difference that you have found in your results. And my question, on that point, I realized that it's very interesting. And my question is related if this sex difference might be with the same P53 mutation, might be related first with the estrogen levels with aromatase activity, and also if in some tumors the immune system might be not well regulated.
Yes. So with regards, yes, there is a sex, it's increased. If there are, in our cohort, is 2.7 girls for each boy. And this is the same in other cohorts, in the group of São Paulo, in other groups in Curitiba, and also in the San Jude and elsewhere. So this sex difference is not, the reason is not known. So there are few hypotheses, but this is something that interests a lot to me, Alicia, you're right. But with regards to this methylation study, when you perform the analysis, you must exclude sex chromosomes in the methylation study. So I cannot tell anything about sex difference here because we do not study the X and the Y chromosome in the methylation profile. This is something that is important to do, otherwise it will be a biased results if you not do that. And yes, the immune system is important in adrenal tumors in adults and in children in regards to HLA system.
Okay, last question, short answer.
Sunir, a tua idade de variação da casuística, ela varia até 16 anos.
Yeah.
Eu vi que os dados do PCAT1, você encontrou uma correlação com a idade. Mas será que uma doença aos 4 anos a gente poderia ter a mesma etiologia? E eu me lembro, primeiro, parabéns, você tem uma linha de pesquisa muito sólida. Você analisou isso com os seus dados passados? Se você aumenta o teu fator preditor, como havia a WNT da tua livre docência?
Você não foi o revisor, né? Você não foi um dos revisores do ERC, não, né? You're not one of the reviewers. Yeah, this question is very important, and we adjusted our multivariate analysis adjusted for age, and even after this adjustment, because age is a potential, is a co-founder, and the significance is still very high. So we are sure that the methylation signature, of course, there may be difference in a child with 4 years and at 16 years old. The methylation is different, but in our data are adjusted, so the difference remains. Thank you.
Bien, el siguiente trabajo será presentado por el Dr. Vergada, Ignacio Vergada. Novel insulin-like growth factor 1 gene mutations, broadening of the phenotype and implication for insulin resistance.
Buenos días. Good morning. No disclosures here. IGF-1 gene mutations are extremely rare causes of pre- and postnatal growth retardation. Phenotype can be heterogeneous with varying degrees of neurosensory deafness, cognitive defects, glucose metabolism impairment, and short stature. This study describes a 12.5-year-old girl presenting with severe short stature and insulin resistance, but with normal hearing and neurological developmental and at the lower limit of normal. This girl was, let's see here. She was born IGOGR at 32 weeks of gestation, conceived by IVF from consanguineous parents. Birth weight was minus 2.0, and she consulted at 3 years of age with a height very low, below minus 4.6 standard deviations, with microcephaly, and the mother was 0.05, and the father was, his height was minus 2.7. Mild dysmorphias like bulbous nose and clinodactyly, as I told you, has no deafness, and mental retardation was borderline. You can see here that at 4 years of age, she was treated with growth hormone up to 8 years of age without any improvement in her height, so therefore it was withdrawn, and puberty started at 11 years of age. Laboratory showed when she consulted at 3 years that IGF-1 was below the range of normal, with 2 tests of growth stimulation tests with normal growth hormone, 20.5 and 23.1. While she was on growth hormone treatment, IGF-1 raised a little bit, but not that much, and then it was withdrawn, and when she was 12.5 years of age, she had an OGGT with insulin intolerance. See that at 120 minutes, glucose was 182, and very high hyperinsulinemia, 28 basal that went up to 558. While she was on metformin, the OGGT improved. Here's the pedigree of the family and a whole exome sequencing study that was done in Italy, showed an IGF variant as the codon 247 that changed serine to cysteine, and according to ACMG classification was a missense likely pathogenic variant. This variant fell on the C domain, and there is only one paper here in the C domain, the one from Nectin. There are very few papers with mutations in IGF-1, and this is the second in the C domain of the peptide. The patient was homozygous and the parents were heterozygous. So the studies performed at our institute were the first, where we can see here that serine is highly conserved along different species, as well as in IGF-2. And the modeling of the protein, we see that the serine falls in the loop of the IGF-1 that is related to the linkage with the IGF-1 receptor. And although the change from serine to cysteine belongs to the same polar amino acids, here the modeling shows that serine exerts a very negatively charged area at this place, and when it reacts with the domain L1, CR, and L2 from the pocket of IGF-1 receptor that is also negative, this might suggest that there is an impairment of binding. So the following studies were IGF-1 receptor phosphorylation assay in the human embryonic kidney cells, HEX cells, that were cultured for 10 minutes with the patient sera, parent sera, and controls. And we can see here in this band that there is very, very little phosphorylation of the insulin receptor, and when the binding proteins were extracted, here's the patient's band, that there is almost no phosphorylation of the insulin receptors. To assess how the viability of the biological activity of this variant, we assess in a cell assay viability, and we see that although there is proliferation of the cells, there is significantly lower compared to controls. The second study was a transfection study with the wild-type IGF-1 gene in the HEX cells and the wild-type variant by site-directed mutagenesis. We selected the better cells that express the protein, and again we see here the cell viability at day 6 cultivation. The variant, the proliferation is much less compared to the wild type. And going to the studies of apoptosis, counting cells, and with the orange acridine test, we can see that the variants cannot rescue the cells from apoptosis, while the wild type does. We know that IGF-1 induces proliferation and inhibits apoptosis. So after that, we thought that the severe insulin resistance could be that the variant might interfere with the insulin receptor. So we go back to this cartoon, and we can see this was the IGF-1 receptor. When we go to the insulin receptor, the pocket is now positive. So probably the variant can induce a ligand with this IGF-1 variant and block the insulin receptor at a certain way. And for this, we perform a study silencing the IGF-1 receptors in prostate cancer cells for 10 minutes. And well, we didn't find what we thought we could find, because with the patient sera, there is some band of the insulin receptor. So the patient sera that has the variant does not compete with the insulin receptor. A further study, a chronic, thinking that might be a downregulation of the insulin receptor, we cultivated the cell for a long period of time, and we had a good expression of messenger and protein of the insulin receptor. Therefore, this is was not explain our theory regarding the insulin resistance. So in conclusion, we describe the second case of partial IGF-1 deficiency due to a missense variant located in the C domain of the protein IGF-1, but the first with an early severe impairment of glucose homeostasis expanding the phenotype. We have explored and described in detail the effect of the variant on IGF-1 activation, demonstrating how partial IGF-1 deficiency might be misdiagnosed due to non-classical phenotype and unreliable IGF-1 assessments. The mechanism behind the insulin resistance remains elusive. Thank you.
Ignacio, I've got a question about this family. This family is consanguineous. The father is minus 2 point something standard deviations in height. The mother is plus 0.5, and they're both heterozygotes. What's happening with this father?
Well, first, the family is in Italy. We don't know the family. The paper is a collaboration with Dr. Giacomosi, but maybe you know. We asked a lot of information about the family, and we couldn't get it. So I understand your thoughts.
Sí, Verónica.
Ladies first.
Verónica Merrick de Chile. Felicitaciones, muy lindo estudio, Ignacio.
Gracias.
Mi pregunta es si tiene composición corporal esta chica, porque los defectos de IGF-1 se han visto asociados obviamente a disminución de la adiposidad general, que es la que induce la insulinoresistencia más que la interferencia propia interreceptor, que es lo que ustedes habían postulado. ¿Tiene composición corporal?
No, pero el desvío estándar de peso era mucho peor. Era menos 8 desvíos contra menos 4 de la niña a los 3 años. Después no tengo axología de más grande.
Y probablemente si tú ahí haces una composición, lo que vas a ver más mermado a lo mejor es la grasa, que es donde tiene la acción insulina, y eso en general es lo que induce la insulinoresistencia en estos casos.
Interesante, gracias.
Can I complete in the same way? These patients can have IGF-1 is very important for pre-adipocyte differentiator, and these children looks like have a lipodystrophy, so maybe they have a very low weight development. Then their insulin resistance. Do you have any data about triglycerides and hepatic steatosis and other?
They were normal, but the patient was born more. Birth weight was not that bad, minus 1.5, and, yes. And the length was worse. But at 3 years, she was really skinny, yeah.
Like a dystrophy.
Okay.
And sometimes in consanguineous individuals, we have 2 different genetic causes in the same patients.
Okay. One of the papers has been under revision by JCM, and we asked to the people from Italy if they look at all the genes of insulin resistance or compared to, and they couldn't find any. Unfortunately, due to the protection of data, we couldn't get all the study of the whole exome to do it ourselves. So it was a good point. We thought about that, but I don't know if we have any answer to that.
Yeah, usually it's more like with insulin receptor, like leprechanism, that have other associated symptoms that your patient didn't have. You know, your patient mainly is a loss of weight and height over time, severe. And compared to the case from Irene Machine that is described for a boy, it was milder compared to yours and responded to a high dose of growth hormone. Of course, in her patient, insulin resistance was present, but was not as severe as yours. So I want to congratulate you, Ignacio. I think it's a beautiful, not only the case description, but what you have done to try to understand at the cellular basis what's going on with this mutation. I think that's very elegant. I think I was a bit more reassured about the insulin pathway after you show that you didn't find the same severity as with the IGF-1, IGF receptor finding.
Okay.
Compared to the question from Fernando, we have seen this before, that although heterozygous parents do have a different degree of impact on height. So I wasn't so surprised about that, but I think it's a very important point. Thank you.
Yeah. Cecilia, that's a minus 2.7 versus.
Yes, I saw, but we had a
two
patient.
standard deviations.
Yeah, we have a patient in Spain that the father was more affected than the mother. So it is important, but the variability, not everybody responds in the same way. So I think it's an important point, as I mentioned, but it has been described in other patients. There are very few of these, so we don't have like extended database to compare it. But I think that what you did for the cellular signaling is really beautiful. Thank you.
Thank you.
Bien, muchas gracias,
Thank
Ignacio.
you very much.
The next speaker is Nayara Dantas from Brazil, and the subject is identification of new genetic modifiers of the phenotype in shock hyperinsufficiency.
Gracias. I have no conflict of interest. The shock satural box contain gene are located in pseudo-region 1 of the sex chromosomes and have a pseudo-autosomal dominance irritant, which needs 2 copies for normal functioning. This gene expressed in groove plate and acts like activation transcription factor in several genes of RUNX2 FGF3. Alterations in this gene are related to idiopathic short stature and laryngeal dyschondrosteosis, but great phenotype variability within even the same family with the same alteration. Here we can see individuals with minus 1.2 with high SDS and individuals with severe short stature, and they can vary also in degree of body disproportion and absence of Madelung deformity. But don't have a genotype-phenotype correlation. What you know is in individual with alteration restricted to the regulatory region, we have less severe phenotype and a lower prevalence of dysmorphic signs when we compare with individuals with alteration encoding region. So how to explain the phenotype variability in SHOX deficiency? If it would be variants in the promoter region, in regulatory region close to the gene, or variants in other genes. So the objective of our work was to evaluate the presence of genetic modifiers that modulate the phenotype in the heterozygous SHOX defects. So we selected 170 individuals with haploinsufficiency of the SHOX, followed up in the Hospital das Clínicas. We phenotyped these individuals with high SDS, sitting high in high SDS, absence or ausência of Madelung deformity, and we selected 98 individuals representing streams of the phenotype and or with alteration restricted to SHOX regulatory region. We analyzed it from a customized panel for gene sequencing, and we had 8 individuals with SHOX coding alteration and 18 individuals with alteration restricted to SHOX regulatory region. We filtered these variants and we selected rare variants with frequency less than 0.01, variants predicting to pathogenicity by in silico tools, and we classifying according to SAMG criteria. This panel had genes related to SHOX, with genes with the retinoic pathway, genes related to growth plate and short stature, the entire SHOX genomic region, including regulatory 5 and 3 regions. According to the results, we found 1 likely pathogenic variant in the CYP26C1 gene and 4 variants of unsettled significance in NPR2, RUNX2, TP53, and ACAN in individuals with alteration in coding region, with a prevalence of likely pathogenic variants of 3%. In one of this family, we have this case of an individual with SHOX and CYP26C1 variant. He had a degree of body disproportion more than as usually we see individuals with SHOX haploinsufficiency. This variant was already described, has a modulator of phenotype in shock appropriation by Montalbano, and had functional studies. In this other family, we had a mother with a SHOX and NPR2 alteration. She had a more severe short stature with high SDS of -4.2, body disproportion, and presence of Madelung deformity when compared to Rodora, who had isolated SHOX alteration. NPR2 variants are already being described associated with a phenotype similar to Lary-Weill dyschondrosteosis. In the individuals with alteration restricted to SHOX regulatory region, we found 3 like pathogenic variants in ACAM, PTA-L8 is CYP25C1 genes, and 1 variant of uncertain significance in NPR2, with a prevalence of likely pathogenic variants of 30% in these individuals. When we compare these 2 groups, we have a high frequency of likely pathogenic variants in individuals with alteration restricted to the regulatory region, with a P equal 0.03. In this family, we have the problem in the father with 2 alterations, the SHOX and a frame shift in a CAM. The problem, in addition to the severe short stature, had advanced bony age, despite being prepubertal, which is common finding in a CAM gene alteration. In this other family, we have an individual with a PTA variant in addition to deletion of SHOX area. This variant is associated with the phenotype of Brachydactyly type A. That is characterized by SHOX metacarpal and SHOX station. Our patient had a shortening for metacarpal in addition to the short station, and he irritated the SHOX alteration from her mother. She had a high SDS of -0.7 without another finds. In this family, we have the index case in the mother with a severe short stature and a SHOX and NPR2 variants. The mother irritated the NPR2 variant from her father he had a severe short stature, and the SHOX alteration from her mother with a high SDS in the lower limit of normality. So here we can see the NPR2 variant segregating with the short stature in 3 generations. In conclusion, the presence of second variant in growth plate-related genes in patients with SHOX appropriation may play an important role in determining the penetrance expression of the phenotype. The potential mechanism of the second variant would be the intensification of chondrocyte dysregulation in the growth plate zones related to the primary alteration of SHOX, leading to a more severe phenotype. The presence of a second-like pathogenic variant is not uncommon in patients with SHOX deficiency, especially in those whose alteration is restricted to the regulatory region of the gene. I would like to thank you to my supervisor, Dr. Alexander Jorge, and the entire team that's running this work with me. Thank you all.
Parabéns. Muy lindo trabajo. Quiero hacer un comentario. En vista de tus hallazgos, ¿sugerirías hacer mejor exoma versus targeted gene en un paciente que tiene signología de SHOX, pero que no terminamos de entender? O incluso pensando solamente en que tiene una discondroteosis por la posibilidad de que tenga otros genes asociados, ¿qué crees que es más valioso?
We did the panel because the cost of exoma, but we have another work running with we doing exoma in the patients that we have finding an individual with SHOX alteration in another alteration showing the exoma. But for this work, we need to use the panel.
I understand that you use a panel, but I'm saying after you see the results, probably now waiting the results that you have found, maybe now you would suggest to the patients to do exome instead of doing a panel. That's what I'm asking. What do you think?
With shocks?
Well, not only with shocks, with ISS or something that you think that they have some Madelung. It could be a shocks, but if they may have also another gene, it would be more worthy to do.
Well, yes, it's a good question because in exoma we can see deletions in the shocks too. So we are capable to identify shocks deletion in shock stature patients with exoma.
Thank you.
Adolfo? No?
Just a short question by curiosity. I know that you studied these second variants as modifiers, but have you analyzed if these second variants had any intrinsic effect in individuals without shocks? Mutations?
This variant, some of that variants has one describing individuals with short stature without shocks, with likely pathogenic variants. This is a good question because in the last family, the NPR2 appears to have a more effect than in the short stature than the shocks variant.
Thank you, Nayara.
The next paper is Differentiated Thyrocarcinoma in Pediatrics, Changes in Disease Stratification at 3 years in a Single Center by Patricia Papendiek from Buenos Aires, Argentina.
Well, hello everybody. I am Patricia Papendiek and will present the work Response to Initial Treatment in a Pediatric Cohort of Patients with Differentiated Thyroid Cancer, Changes after 3 years of Follow-up. Childhood onset DTC has a more aggressive presentation, but no overall increase in mortality compared to adults. The objectives of our work were in first place, to characterize a pediatric cohort with DTC at diagnosis using the American Thyroid Association Initial Risk Stratification, IRS, following initial treatment. Patients were categorized in 3 groups according to histology, serum thyroglobulin, TG, and serum TG-AB, and whole body scan. Low-risk patients were those patients with intrathyroidal tumor, no lymph nodes or incidental level 6 lymph nodes, with complete resection, no invasion, and no aggressive histology, while intermediate patients were those patients with intrathyroidal tumor and extensive level 6 lymph nodes or minimal lymph nodes outside level 6. High-risk patients were those patients with local invasive disease, extensive lymph nodes outside level 6, with or without distant metastasis. The second objective was to evaluate the response to initial treatment until 3 years based on the modified ARTA Dynamic Risk Stratification, DRS, for adults. Clinical outcome or disease status was assessed yearly according to serum TG and TG-AB levels and images into 4 response categories. Excellent responders were those patients with negative TG or TG-AB and negative imaging. Indeterminate patients, responders were those patients with low levels of TG or ATG and non-specific findings on imaging studies. And there were 2 categories of incomplete responders. Biochemical incomplete response with those patients with high levels of TG or ATG or increasing levels with negative imaging, and structural incomplete response were those patients with structural or functional evidence of disease. Of a total cohort of 51 patients with DTC followed at our institution between 2008 and 2019, 31 patients with complete data and the minimal follow-up of 3 years were included in this retrospective study. Clinical pathological data and outcomes were analyzed. Until 2015, patients were treated initially with total thyroidectomy and radioiodine ablation, while after that, following the new ATA guidelines for children, low-risk patients were accepted from radioiodine ablation. Demographic data of the whole cohort are summarized in this table. Median age was 13.7 with a range from 5 to 18 years. Predominant sex was female. 80% of the patients were already pubertal at diagnosis. And regarding the histology, most of the patients had a papillary thyroid carcinoma, while only one patient had a follicular thyroid carcinoma. The classic variant PTC was the most frequent variant, followed by the follicular variant and the diffuse sclerosant variant. 2 patients had a double thyroid carcinoma. About half of the patients had a multifocal tumor. Median maximum nodule diameter was 18 millimeters. 16% of the patients had already distant metastasis at diagnosis, and 35% of the patients had also thyroiditis. In this figure, you can see how each initial risk group behaved individually during the study period. So, initial low-risk patients remained free of disease all over the study period, while intermediate risk patients achieved 86% of excellent response at 3 years. High-risk patients achieved 36% of excellent response at 3 years, about 30% an indeterminate response, and the remaining third of the patients persisted with disease. Only one patient of the intermediate risk group presented at 2 years a cervical relapse. We compared initial high-risk group patients with incomplete response, those with biochemical incomplete response and structural incomplete response, with those who achieved an excellent response, and found that patients with incomplete response at 3 years were initially predominantly prepubertal, with a higher proportion of T4 stage tumors and distant metastasis than those with excellent response. So, in conclusion, our data confirm the aggressiveness of pediatric PTC at presentation. Clinical outcome by DRS at 3 years showed a marked change in the cohort risk composition compared to admission. Low-risk patients remained free of disease, and intermediate patients showed a trend to a better outcome. Half of the high-risk patients behaved promptly towards a more benign status, while in the rest, improvement was slower or still incomplete. The early DRS assessment provided us useful information, allowing a less aggressive approach to those that evolved to an excellent response. Although these observations have to be extended, they reassure the therapeutic intervention. Thank you very much.
Thank you, Patricia. I've got a question about the 3-year kind of lapse at which you're looking at this. Do you think that this might change if you look at them 5 years after intervention? Talking about the interval that you're following them, it's relatively short. Is it possible that things may change as the patients get a little older?
May I respond in Spanish?
Sure.
Okay. Primero quiero aclarar que en esta presentación falta una diapositiva, pero bueno, después en realidad marca cómo es la composición del riesgo a lo largo de los 3 años. No sé por qué falta en la presentación. La pregunta es si a los 5 años esta composición de riesgo y la respuesta al tratamiento inicial sería diferente. Probablemente sí. De hecho, los pacientes de alto riesgo, que son los que realmente no cambian tanto a los 3 años o que tienen menor cambio a los 3 años, continúan el tratamiento de acuerdo a su estratificación de riesgo anual y continúan recibiendo eventualmente tratamiento con yodo radioactivo, por lo que es probable que estos pacientes en los años posteriores alcancen una mejor respuesta.
Sí, la pregunta un poco tiene que ver con los de riesgo intermedio, que son los que se ven más o menos bien y que uno espera que sigan bien y que no tengamos alguna sorpresa a los 5 o 10 años.
Es difícil de prever, pero en general responden bien, ¿sí? No tengo los datos, justamente esto tuvimos un trabajo previo que comparábamos los pacientes a los 2 años y en esta cohorte son pacientes que hemos seguido y comparado los resultados de esta estratificación de riesgo a los 3 años y los resultados muestran que los pacientes aún con un N mayor se comportan de la misma forma y alcanzan una mejor respuesta a los 3 años.
Ya.
Esto es un trabajo a continuar el trabajo con estos pacientes y ampliar el N.
Bueno,
Hugo. Excellent, Patricia. Would you like to make my question in Spanish or in English?
In Spanish, please.
Spanish. En vista del alto porcentaje de pacientes con tiroiditis autoinmune que vos mostraste en tu serie y la interferencia de los anticuerpos antitiroglobulina, que como fue aclarado hoy temprano, nos complican el marcador tumoral, en adultos ha sido propuesto seguirlos con el título de anticuerpos antitiroglobulina, porque en caso de que permaneciesen constante, podría ser como que no hay resto tumoral que esté aumentando anticuerpos y entonces uno pudiese quedarse un poco más tranquilo validando la tiroglobulina. ¿Alguna vez has probado usarlo o lo hicieron en esta serie?
Sí, de hecho nosotros en el servicio cuantificamos los títulos de anticuerpos antitiroglobulina y eso nos permite evaluar cómo es la curva de descenso eventualmente, sobre todo conociendo que los pacientes con tiroiditis y anticuerpos antitiroglobulina positivos pueden persistir con anticuerpos positivos a lo largo de hasta 3 e incluso 5 años post tratamiento inicial. Así que sí, es algo que normalmente hacemos.
Y en ese caso, si aumentasen los anticuerpos, sería motivo de preocupación.
Por supuesto.
Ya, última pregunta, Alicia.
Muy lindo el trabajo, Patricia. Yo te voy a hacer en castellano. No, la pregunta es que en tu cohorte la mayoría de los pacientes eran pobres. Y uno sabe que los chicos más grandes tienen mejor pronóstico. Y mi pregunta es, tomando un poquito lo de Fernando, si vos, si uno tuviera un estudio en una población de niños más pequeños, la pregunta es si el tiempo de seguimiento debería ser mayor cuando vos por riesgo dinámico lo estratificás intermedio, porque uno sabe que el pronóstico probablemente no sea tan bueno como en las poblaciones más grandes. O sea, me llamó la atención que sean todos púberes. Hay
80% de pacientes puberales. El grupo de pacientes prepuberales de esta cohorte no es alto, ¿sí? Pero claramente fueron aquellos pacientes que respondieron peor al tratamiento inicial y estaban incluidos entre los grupos de alto riesgo. Si estos pacientes requieren mayor seguimiento a largo plazo, sin ninguna duda, son pacientes que requieren un seguimiento a lo largo de toda la vida, ¿sí? Habría que ampliarle el N para conocer y ampliar el N de pacientes prepuberales para poderle darle una respuesta a lo que usted me plantea.
Bien, muchas gracias, Patricia.
Gracias.
El último trabajo es de Juliana Moreira Márquez, Generating Pituitary Cell from Induced Pluripotential Stem Cell of Patients with Child Onset of Congenital Hypopituitaris.
Good morning, everyone. First of all, I'd like to thank the organizing committee for the opportunity to present our study here. So this study is developed by the University of São Paulo Medical School, which is the place where I come from, in a partnership with the University of California, San Francisco, where I'm in right now to do part of the experiments. So induced pluripotent stem cells granted the Nobel Prize in 2012 to Yamanaka and Gurdon, which were the ones who discovered that somatic cells, for example, blood cells or fibroblasts, could be reprogrammed using viruses or plasmids containing pluripotency genes into pluripotent stem cells. And these pluripotent cells can be differentiated into any cell type in the human body. For example, cardiomyocytes, hematopoietic cells, pituitary cells, neurons, which can later be used for further studies such as disease modeling, drug screening, and autologous cell therapy. So why do we need induced pituitary cells? So first of all, we need these cells because pituitary is an accessible tissue as well as the brain and the heart. And also, we lack studies using human cells, as most of all what we know for now, it is basically using animal models. And also, in a disease-based, patients with pituitary diseases such as hypopituitarism, they have to go through a lifelong treatment. And also, congenital cases of hypopituitarism, they have a very, very low diagnostic rate, molecular diagnosis rate, which ranges from 15 to 20% in most of the cases in the worldwide cohort. And also, even patients with the same gene affected, they have a very high phenotypic variability. So the first aim of my study is to produce pituitary cells and then characterize the cells using pituitary-specific factors such as pituitary transcription factors and also hormones. And the second step is to do a hormonal secretion assay to determine if these cells are able to produce hormones and also if they are able to respond to hormonal stimuli. And the third part is to do an in vivo functioning assay to understand if these cells are able to restore the capability of producing hormones in models of hypopituitarism. Okay, so how can we make the cells? So to make these pituitary cells, we need to resemble the pituitary embryogenesis, upregulating genes that are associated to pituitary development, such as LHX3 and PROP1, and also genes related to mature pituitary cells, such as the hormonal markers. So to do that, we mimetize the signaling molecules that also occur naturally during pituitary embryology in humans. So we use BMP4 signaling, FGF8 and FGF10 signaling, as well as sonic hedgehog. And also we add to the basal media some non-pituitary cells inhibitors, such as SB, which is a TGF-beta inhibitor. With that, then we generated a transcriptional map from different time points of the pituitary. So the first column represents the cells that were not induced, and we saw an induction of most of the pituitary transcription factors throughout the times of the differentiation, especially from day 10 to day 15. So higher expression here is represented by lighter colors, while darker colors represented lower expression levels. So for the pituitary hormones, we also saw a higher expression in later time points, especially for GH, ACTH, and TSH, but LH beta and prolactin were also induced. We confirmed the transcriptional levels using protein assays. So the first one is a high-throughput imaging quantification, and we confirmed that LHX3 is activated during the time points that we analyzed, as well as ACTH and GH. So we can see in the top right image, LHX3 expressing cells, as well as ACTH cytoplasm for the cells that we produced. And also, we determined that by the entirety of differentiation, we have about 60% of the cells in our culture corresponding to pituitary cells. And we can also see other markers of pituitary, such as PROP1 in the right lower image. Besides GH and ACTH, the cells they are also able to produce other pituitary hormones, such as TSH, LH, and FSH. And they keep producing hormones until later stages of differentiation, such as day 120. As we see here, we have GH expression as well as ACTH expression at day 120. We also performed stimuli assay using CRH. So we added CRH for 20 minutes to the cell media to see if they were able to respond and to increase their ACTH production. And we saw increase according to the different dosages of CRH, which was statistically significant. And we also see positive cells for ACTH increased in the induced cell lines. The second aim of my study was to understand if we could use this method for disease modeling and if this would represent in vivo phenotype. So we generated patient cells, patient iPSCs, and then differentiated the cells into pituitary to see if they resemble the disease in vivo. So we used two patients which were siblings. They had two compound heterozygous mutations in the PROP1 gene, and they were diagnosed with hypopituitarism during childhood, with GH, TSH, and LH and FSH deficiency. And the second patient was later, during adolescence, diagnosed with ACTH deficiency. And we used their parents as the control for the experiments. So we collected blood from these patients, and then we produced the pluripotent stem cells, which were effective, as we see in the right graphs, because we have more than 90% of cells positive for all of the pluripotency markers, such as OX2, NANOG, and OX3 and 4. Not going, I'm sorry. Oops, yeah. So these cells were able to resemble the in vivo functioning as we see in the PROP1 expression level. So the patient cells, during all of the times that we analyzed, they had lower PROP1 levels compared to the control cells. And we also saw lower GH1, TSH, and LH levels in the patients and normal PONC, as the patient that we analyzed didn't have ACTH deficiency. So we saw normal ACTH secreting cells in the patient cell lines, but lower levels of GH and TSH compared to the control cells. So we can consider that we can make pituitary cells in a dish. These cells, they respond to stimuli, especially for ACTH, which was the one that we tested. And the CPHD patient cells, they resemble the in vivo phenotype in vitro. So we can use these cells for better understanding of pituitary development, function, and also disease. I would like to thank you all, and also my collaborators and lab members.
Okay. Do you think this kind of studies can demonstrate the modification according the age in patients with hypopituitarism? I mean, most child has not hypothyroidism at the moment they have growth hormone deficiency. So how we explain in this model the late manifestation of the hormone deficiency?
So this is something to be stated so when we differentiate the cells, we don't know which time point from the human development we are taking. So this is studies, especially for pituitary, because it's a very, very new area that we are using the cells for. We have to establish when we have, for example, puberty, childhood, or adulthood. We can guess from cell characteristics, but we need further studies to see that. And then we can determine the hormone levels based on that.
Juliana, very interesting paper. I've got a question about the possibility of selectively inducing the development of specific cells within the pituitary. Let's say, can we, with this kind of technology, get closer to, for example, inducing the development of somatotropes instead of the other cells? How difficult do you think that might be?
Sure, we have two different approaches in order to do that. So we can use the embryonic development signaling, for example, adding more FGF8 to induce some cell types in the pituitary, or more SHH for other cell types. And we also have manipulation of the cells right now, where we can add plasmids with these genes related to the cell of interest. For example, tyrotrophs, we can add genes related to tyrotroph differentiation, as well as for other cell types in the pituitary.
Okay, thank you very much. Y a todos ustedes, muchas gracias por su asistencia. Pasamos a la siguiente presentación. ¿Hay un break ahora? Pero muy breve, o sea, sí. Casi salen y regresan.
We need everybody to leave the room and come back later. Is that right? For the symposium, you have to leave the room and come back for the symposium. So everybody out.